wild type is, okay, so I'm hearing most typical, most common, most normally found.
So isn't wild type just what's naturally found? I feel what's common.
What's naturally found, what's common.
But could we say that previous ancestors of the Homo sapiens had shorter fingers?
But not necessarily. We're talking about modern humans. So brachydactyly is a genetic, it's caused by a genetic process. There's an allele of a gene. You get one allele, you get long, what we call wild type brachydactyly, or finger length, phalanges length. And there's a version of this gene that causes brachydactyly. So who's the most wild type human?
Right-handed, male Chinese.
Right, according to this video, which I'm not trying to say that that is true, but I think it's an interesting video to spark this discussion. Is the most common or the most wild type version of a Homo sapiens a 20-year-old male Han-Chinese right-handed person who has no bank account and makes whatever amount of money? $12,000 or less a year. You can translate that into yen if you want. So, does this concept of wild, how do we use the concept of wild type? When is it okay and when is it not okay? Or when is it not valid to use this concept? Thoughts?
You only compare two?
That's a good start at the very least. When you've got two different things and you're saying which one is maybe more common, you would call that the wild type. And the one that's less common or rarer, you would call that the, not necessarily a mutant version, but you would call that a different version. And of course, how many most common, as this picture shows, how many of these individuals are on the planet? I don't remember the exact number.
7?
No.
9 million?
9 million.
Okay. How many people are on the planet? It's in the billions. So even if this is the most common phenotype, which is what this movie claims, that doesn't make that demographic or that population the majority. That's why that quote there is really critical. Typical is always relative. So you have to say, it's wild type in this context. So you might say, you're looking at a population of western pond turtles. And in that population, here's the wild type trait, here's the mutant trait. But if you look at other populations, that all goes out the window, potentially. So anytime we use the term wild type or mutant, we have to be very particular about the terminology to define what population we're talking about. This will become very important in a second when we start working on a question.
Can something change from wild type, or from mutant to wild type if it gets enough of the population, I guess?
Right. And that's where the term mutant comes in, is a little bit difficult to work with. Allele 1, let's say that allele 1 we'll always call the wild type allele. WT. And let's say there's a second allele and it's less common. So we'll just say it's rare or rarer. It doesn't necessarily make it mutant. It's genetically different. If you remember back to when either a movie or in class, I don't remember which, because my brain is scrambled, talked about the idea of alleles are two different versions of a gene. And the only way that a geneticist knows that there are two different versions of a gene is because they are different at the sequence level. So at the end of the day, alleles are defined by the actual sequence of the DNA. So you might have allele 1 might be some random DNA sequence. And allele 2 is defined as being allele 2 because there's a change right there that distinguishes those two versions of the gene. If you're going to be talking about alleles, it's going to be due to a genetic difference, a DNA sequence difference between the versions of the gene. That's how we know that there are two alleles. If it was the same DNA sequence, there would be no difference, and so there would be no difference. But you're absolutely right. So if the allele 2 version of this gene became more prevalent over time, then that would become the most common version of the gene, and that would become the wild type allele in the population, in the entire species, on the planet, in the universe, whatever scale we're looking at, whatever population we're talking about.
Or maybe in terms of finches on that.
Right, or maybe in terms of Darwin's finches, or, ooh, by the way, speaking of Darwin's finches, I just saw this in my mailbox. Dr. Neil Shubin, Your Inner Fish. Anybody who's really interested in human evolution should go see Neil Shubin talk. He's an internationally renowned biologist. He works at the University of Chicago, but he's going to be at Fresno City College giving a seminar, and you can get more information from me. I'll post this on Blackboard. That's for any of you that are heading towards 105, Biology 105, especially if you're evolution bound, if you're a biology major, you should go check this out. It's really interesting. He's a fish biologist, but he's most famous for having found the quote-unquote living fossil, a fish that seems to be a transition between fish and terrestrial invertebrates. So it's really interesting. He's a really good speaker. Sorry, brief distraction. Now, wild type versus mutant. Do you have any questions, clarifications, concerns?
So it's just at a given moment, what is most common?
It's, yes. If you want to put this into practice, it's what's most common at that time in that circumstance. Even if it's 51%. Even if, how many different alleles of a gene can there be? I think we might have had this conversation before, but I want to make sure that we've had it at least once. This is not an answer that has not a question that has a right answer. So take a stab at it. How many different versions can there be of a gene?
I don't want to say infinite.
Okay, fair enough. I don't want to say infinite, but close. Don't do any math. I see some of you trying to do some math in your heads. Give me qualitative. Yeah.
Would it just be relative to the gene, how many mutations can be observed that make it different at some level?
Basically, that's it. So you could say, if we're talking about, like I drew up here, an 8 nucleotide long gene, you could figure out, I don't want to do the math right now. I don't ask you to. You could figure out how many different single genetic differences there could be. 4 nucleotides per base, 8 bases. But so the point is, when you this is not the normal length of a gene, people. This is convenient for me to draw on PowerPoint slide. Genes are hundreds or thousands of nucleotides long. So in essence, there could be an essentially infinite number of different versions of a gene that exist in the world for any gene for any species. So if you think about the human gene that encodes DNA polymerase, the enzyme that replicates our DNA, one of the genes we've talked about in class before. There are lots and lots and lots and lots and lots of different versions of alleles of that gene floating around the human population in different individuals in different parts of the world. And those change over time. So if you're talking about one population, one version of that gene could be more frequent. The point is, there's never a majority. Rarely will there be a majority. So if you think about, say there's 10 alleles of a gene. You've got allele 1, allele 2, and so forth, down to allele 10. It's likely that there's not a single one of those 10 versions of that gene that are the majority, 50% plus. So it's rare that wild type is, what would you say, most common.
So there's no such thing as, wait, so what you're saying is, when you say that there's never a majority, so we never know exactly which one is which, but we know that most of them.
So it's difficult for a biologist to know which allele has the highest frequency. That would mean sampling every human in the world, for example. You'd have to take everybody's DNA and calculate exactly what frequency of every allele is. The easiest thing to do is to say, and this is just how we do this in practice, that there are more allele 1s than allele 3s. So in that pairwise comparison, allele 1 is more wild type. We call it wild type than allele 3. Yeah.
One thing that I've noticed is that we haven't necessarily talked about dominant and recessive. So
Totally different concept. There's a reason why we haven't talked about dominance and recessiveness yet. Go ahead.
in terms of what we're talking about today, if there is one that is dominant, it could essentially become the one, right?
Indeed. So here's, before we move on, we're gonna move on to talking about Punnett squares and crosses again in just a second. But when we pick this back up Monday, I want to have already given you this information in advance. What does this mean? If you remember anything from this class, that's going to be a key concept.
It's not the most common.
Dominant means something else, but it doesn't mean dominant traits are not always, there's no connection between what's most common and what's dominant. So what's the most common hair color in this room? Brown. Not necessarily dominant, it's predominant. It's common in this classroom, but that doesn't necessarily mean that when you get down to the DNA level that the enzymes that make the brown pigment are dominant to enzymes that don't make pigment. It's actually likely that they probably are. Maybe it was a bad example. But just because something is common, just because a trait is common does not mean that it's caused by a dominant version of the gene. We'll talk about that more on Monday, but I wanted to plant the seed of that in your minds before we get there. So, wild type is relative. If you're comparing two alleles or more, the one that's most common, this bottom line, the one that's most common you can call wild type. And then it's up to you to what you decide to call the rest. Mutant, different, varieties, variations, etc. So we left off last class talking about, we're finally going to do this Punnett square. How do you make these green fluorescent roller worms? We're going to see which way is the most efficient to do this. And this is the first time I'm going to have done a Punnett square like this for you explicitly, so I wanted to be able to walk this through with you in person in case you have got questions about how this works. So one parent was a roller, but didn't have a GFP gene. The other one was wild type. And a heterozygote, if I remember correctly. Did I get that right? Is that the genotype that we had been using? So we have here a roller. Is that green? Is that close to green? Okay, good. Excellent. Thanks for your help, non-colorblind people. How many people in here are colorblind? Anybody want to claim? Oh, fair enough. So there's a couple of us. So is that the wild type or the mutant condition? In our population, that's the mutant condition, right? The mutation that causes color blindness or the inability to see some colors or many colors is not common. It's pretty rare. So last class, we ended with, what are we going to do with these worms? We have these worms, and we can cross them together. And that roller parent only makes one type of gamete. It's got a minus at the roller gene and a plus at GFP. And the other parent is plus, sorry. getting distracted. It's plus, semicolon plus, so plus at the roller gene and a plus at GFP. The other type of gamete carries the GFP gene, the asterisk. We get that by making all the different combinations. So the genotypes in the middle, and then we ended last class trying to figure out, well, what are we going to do with these offspring? What's the phenotype of this offspring on the left? Phenotype. What do they look like? The top one is the roller locus, the bottom locus is GFP. So you've got one copy of the roller mutation. Are you a roller or are you wild type? Wild type. You had to have 2 minuses to become a roller. So this is a wild type worm that's green fluorescent or not? Not. No asterisk. So it doesn't have that gene that produces the green fluorescent protein. So that's a completely wild type worm. And then its sibling there on the right, what does it look like? It's a green, it's got 1 asterisk, so it's green, wild type. So it's only got 1 minus, not 2 minuses at the roller gene. It's got 1 mutant version, 1 wild type version, normal, common, whatever you want to call it. So neither of those worms shows that trait that we're looking for. Roller, it's green. We're trying to get minus over minus, asterisk over asterisk. That's a worm that will, when you self it, is pure breeding. It only produces individuals with the exact same genotype. So any totally homozygous genotype will be a pure breeding genotype, by definition. So we're going to self this dihybrid. It's green, but it's wild type for the roller locus. Anybody have any concerns at this point? Questions? That's what's coming up next.
So we only want to self that one because it has an asterisk in one and a minus in the other? I'm not going to put this on everybody. That's what somebody suggested last class that we do. Was because we have one version of each of those alleles that we're trying to combine, we're trying to make all minuses and all asterisks. Well, at least we've got one of each in that individual. So that's maybe the closest we've come so far to making the genotype down at the bottom that we're trying to get to. And the question ultimately said, what is the most efficient way? So the way I'm going to suggest that we proceed, the way I'm going to tell you we're going to proceed, is we're going to try what was suggested. We're going to try selfing this worm. If we can make, by just doing one cross with that worm, if we can make this genotype at the bottom, by definition, that's kind of the most efficient way to do it, right? It's a single generation. So there's no way to make a worm that's this genotype faster than that. You have to do at least one more cross. So if selfing gets us there in one generation, that's a good answer. No way to make a worm faster than a generation, unless you happen to know some 3D printer that can print living organisms. So we're going to self a dihybrid. There's a big Punnett square. That's the full 16 square Punnett square that I said I would never make you draw out on an exam, which is true. So let's just set it up. Practice. Because I will ask you to do things like set up big Punnett squares. I just won't ask you to fill it in because that takes so much time. And it's often really easy to make a mistake when you're filling across and filling down and 50 minutes and stress, and not worth it. So let's just set up what's labeled across the top and down the side. The gamete haplotypes.
And this is, because this is a dihybrid, what do we know about what we're gonna write across the top and down the left side? This is selfing. So they're gonna be the same. Whatever we write across the top is gonna be the same down the side. That's the nice thing about selfing, to you. So we're gonna have the first allele with the first allele, so a plus and a plus. That's what's gonna be in one sperm or one egg. Then what else? List it off for me. So then plus with asterisk. Okay. Then minus with plus and minus with asterisk. And we write the exact same thing down the left side. It's the same parent mating with itself. Think about that for a while. And then all of its gametes will have the same ratio. A quarter of each of its gametes will have each of those 4 different combinations of alleles from the GFP locus gene and the roller gene. So we don't need really to fill this in at this point. We should already be able to figure out if we can answer this question. Do we get the genotype we're looking for? We're looking for minus over minus, asterisk over asterisk.
The bottom corner, bottom right corner.
Yep. So that requires being, if you're going to generate that genotype, what would the gametes that create that genotype look like? What's inside those gametes? What are the haplotypes that are going to fuse together to produce that genotype?
A minus and an asterisk.
Yeah, it has to be a minus and an asterisk in both of those gametes to make that genotype. And that's where that intersection is. Right, a minus and an asterisk combined with a minus and an asterisk. Yeah, please.
So when they're combining, are we taking one from that side and one from that side and bringing them together?
Exactly right. So that's the concept of, that's why the Punnett square, again, is a square. On one side is sperm gametes, for example. Sperm, it doesn't matter which. Side, top, you decide. It doesn't matter. One side is male gamete. One axis is female gamete. And this diagram is just representing all the possible ways that they can come together at fertilization. And that's why we fill across and we fill down. It's all the different possible combinations of different chromosomes that can come together in all possible combinations at fertilization. So a 16th of the offspring, we'll get the genotype we're looking for. It's only a 16th of them. It's a 16th of the total area of the square. But we're there. One cross, selfing.
So how do we know it's not gonna be an asterisk and a minus?
So, which one?
For the last one that we just put minus, minus, asterisk, asterisk, how do we know it's not gonna be the minus with the asterisk?
Two different alleles.
Right, so this is that's a roller allele, that's a GFP allele. You could use letters if you like.
Different letters so you don't get the pluses mixed up.
Yeah, that would be more useful. I like the suggestion. So if I had thought ahead more about this, yes, we could use letters to represent the genes instead of just pluses and minuses. In this case, the first character is always which gene? The first character is always, the first symbol, the first letter, the first symbol, is always what we're talking about, the roller locus, and the second one's always GFP. That was by definition on the previous slide. But you're absolutely right. If we didn't have that sort of nomenclature, we wouldn't know which plus and which minus we were talking about. So these are all roller alleles. The first one's listed. And the green ones, the second one's listed for every gamete haplotype or all the GFP alleles. Yeah.
So to check for understanding.
Yeah.
We only mix the roller with the roller and the GFP with the GFP. Because I know what she's asking was that, because I wrote it out too. I got a negative and then an asterisk and then an asterisk and a negative and then an asterisk asterisk and a minus minus. So we don't have to do the minus asterisk.
Yeah, you have to keep the alleles for different genes separate.
Oh, okay.
Yep. And that's only because, and that's only for the present case, you could imagine having two different chromosomes. And those come together in different combinations inside the gametes. So you could have roller plus. Now we're getting to, let's use letters to indicate, or words to describe the genes. So for example, this would be a case where you've got an individual who's heterozygous at the roller locus and homozygous for GFP, 2 asterisks and then a plus and a minus. The way I had been writing that was plus over minus colon asterisk over asterisk. So that's how to translate between what the chromosomes look like and what the notation looks like. And if an individual that looks like this goes through meiosis, you're going to get half of the gametes have the plus and half of the gametes have the minus at the roller gene. And what happens at GFP? You still get, yeah, they're still all going to have an asterisk. But that's only because the individual is homozygous. They can only put asterisks into their gametes. Even though half of those GFP genes, half of those GFP alleles go into one half of the gametes, and the other allele goes into the other half of the gametes. And that's how we get those haplotypes. And that's what goes along the edges of the Punnett square. So in my nomenclature on the previous slide, I would have just called that plus over asterisk, or plus semicolon asterisk. And that would have been plus semicolon asterisk.
This semicolon is the same,
Semicolon means on different chromosomes.
okay, the backslash.
Slash means 2 copies of the same chromosome. Okay. So that's at the top here, roller plus and roller minus, and that's GFP asterisk, asterisk. You've got 2 chromosomes there that have GFP on them. Slash indicates we're talking about 2 separate pieces of DNA, but the same gene. The semicolon is what separates different chromosomes. So we talk about genes on 2 separate chromosomes, then we put a semicolon between.
That would be where individual sort comes in. And if they're on the same chromosome, the only way you're going to get something like that would be through crossing over.
Talk about, yes, next class we'll talk about what the notation is, again, for genes on the same chromosome. Yes.
Dr. Ross, can you see the previous slide really quick again?
Thank you for pointing out that I was not recording. Okay. So the D allele we can figure out. What about C? Do we know anything about?
They both have a
There's
C2.
for sure C2,
Okay. So what's the pattern that's emerging here? When the kids have a certain genotype, we can figure out content of the parents. It's when the kids are homozygous. If there's a homozygote child, then you know that each of the parents had to have one of those alleles. Okay. So we know each of the parents have a C2. That's the only way to get that offspring's genotype, C2 over C2. Had to have a C2 in each parent.
one parent has another C2 and the other one has a C7.
Okay, I heard one parent has a C2 and another parent has a C7.
I can't say both of them have the C7?
Or, so now we've run into the we can't distinguish between the two. Because in either of those cases, the red and the green, you could generate all 3 of those offspring genotypes at the C
Well,
locus.
following that logic, you can have one parent that's D1, D1, the other parent that's D2, D2, D2. So you
No, you
Oh,
can't.
no.
Yeah.
Never mind.
So if you did, no, that's fine. I'm glad you said that. So could you have a parent that's D1 over D1 and D2 over D2? In that case, all of their offspring would be heterozygotes. They'd be D1 over D2. So it's all a big logic problem. Emphasis is that 2 points. One, there are not usually one right answer to these sorts of problems. What would help? Think about that for a second. What would help us answer this more precisely or more confidently? The second point is that when you have homozygotes, those give you more information about the parent genotypes than you have kids that are heterozygous. It's basically meaningless.
If there was one more child that was a C2, C7, D2, or sorry, if it was
You have to be C7.
any C2 combination and then D1, D2 again to fill in like the last square, or fill in another part of the Punnett square, could that help statistically give at least a better reasoning towards what would happen?
So I'll leave that to you guys all to think about. The key concept here is we want humans that have more kids, if this is the case of a human, for example. Yeah, don't tell anybody I said that, except I recorded it. Oh, good, it's still recording. Great. Human geneticists like working with human pedigrees that have large families because how would we distinguish between whether or not this is C2 over C2 or C2 over C7, for example?
We can't, right?
You never can.
You have C7 over C7.
Right. So this is going to get a little bit nebulous if it's not already. I apologize. The only way to know if two parents are both heterozygous is what? So far we've seen C2 over C2s, and we've seen C2s over C7s in the offspring. There's P0, there's F1, bless you. The only way we could guess, if the parents are dead and we don't have their DNA, we can't actually get the DNA and look at it, analyze it. What's the only way to prove that both parents are heterozygotes? You'd have to find what genotype that would prove that both the parents had at least one C7 allele. You'd have to see a kid that had that genotype. Remember, the homozygote is the only one that tells us something about both parents at the same time. So the question then is, how many kids would you have to have before you knew for sure, without a doubt, that that kid was going to exist?
At least 4.
So there's a statistical argument we're going to have to make, and that's something else we're going to talk about when we meet next week, chi-square analysis, and figuring out how many kids do you have to look at before you know statistically significantly that the parents probably don't each have a C7 allele. Because you could have 20 kids, and by chance, you could have both parents have C7 alleles, and just by chance, those gametes never fuse together to produce living kids that you've got in your pedigree. By chance. There's a chance, it's a really small chance. And the more kids you have, the smaller that chance gets. And that's why we have to use statistics next week when we talk about these sorts of things.
Why, like, as a geneticist, why do you care? Like, what do you use this research for? To, like, oh wow, they're C2 over C2. Like, what do you use the research for?
So the main concept here, pedigree analysis, is mainly for human diagnosis, right? So to identify, and again, we're almost there. Thank you, that's a very prescient question to ask. Trying to predict, for example, or to identify what chromosomes are certain genes on, are they dominant or recessive? We can use all of this pedigree information in trying to figure out genotypes to figure out inheritance patterns of diseases. That will tell us next week if they're a dominant inheritance or if they're recessive alleles. And if they're on certain chromosomes versus others, that would be autosomes versus sex chromosomes, X or the Y chromosome, and so forth. So applications coming up, but that's always a good question to ask. So how does this apply to you? How does this apply to society? How does this apply to life? So I hand back here first, and then.
I just wanted to make sure that I understand this. So the reason why one parent, we know for sure should have a C7 over C7 is because we already saw the C2 over C2, which is a homo child, right? And then the C2 over C7 is a hetero.
Yep.
Therefore, the third child or one parent should have C7 over C7.
The only way we know that each parent has a C7 allele is if you see the offspring that is homozygous. Because the only way for them to have the same allele is one had to come from one parent, the other allele had to come from the other parent at fertilization. And the same is true for this kid over there. So ideally, a geneticist would love to see kids that only have homozygous genotypes. That would tell you everything about the parent genotypes. That doesn't happen, of course. Ideally, that would be nice. So the heterozygotes in the middle don't actually give us that much information about the parent genotypes. We don't know anything about which parent has which allele. We just know that one parent, don't know which, has C2, but whichever parent has C2, the other one definitely at least has one C7, right? I just confused you. I confused myself. It's pointless. Right? Heterozygotes don't tell us anything about parent genotypes. The homozygotes is where it's at. Okay. We do this one relatively quickly. This was by email. Thank you very much to the student that sent this in. Good question. Is it possible to do this cross? Stop shaking the video projector. Everyone's going to get seasick. Maybe it was a tiny earthquake. Is it possible to do this cross? And if yes, then is this a test cross? So, I see a hand.
The first thing I just think of is if assuming these are all on different chromosomes, certain species, if you have the reason we can't do it is because they're different number of chromosomes, the fertilization wouldn't
So, okay,
survive as a child.
slashes and semicolons. So how many chromosomes are we talking about? So we've got 3 different molecules of DNA there. So they're separated by semicolons. So we've got 3 sets of chromosomes. A chromosome that's got gene A, a separate chromosome that's got gene B, and a third chromosome that's got gene C. That's what I take out of that notation that the student wrote in this question. So that's one individual, and they're 2 alleles, so that means they're diploid. So you've got an A1, you've got an A2. You've got 2 chromosome Bs, they're each B1, and you've got 2 chromosome Cs. So can you set up this cross? What's going on with the genotype on the right side? Right, the C chromosome isn't specified. Does that mean, so my question, which will probably go unanswered, is the C chromosome there and we just don't have the information, or is the C chromosome missing? Let's assume that it's there. What I'd like you to consider, based on what we talked about at the start of class, and what I've mentioned in previous classes, if you don't have genotype information in any sort of genetics question, you can do one of 2 things. You either, what's the assumption you should make? If it's not stated, it's wild type, so then based on the conversation we had at the start of class, how are you going to decide which allele is wild type? You are welcome to assume, and especially if you ever have to make an assumption, just write it out. Say, assuming this is true, then this is the answer. That's totally fine. So in this case, what I promise you is always going to work, is to say that the first allele, whatever is numbered 1, is most common. So just say, assuming that allele C1 is wild type, what would the genotype on chromosome C be? It would be C1 over C1. You always assume, it's a valid assumption. You get the wild type genotype, homozygous wild type, for any locus that's not explicitly mentioned. Now, of course, that's not necessarily true. It's just the assumption. It's a good starting point. You're welcome to do that.
But if it wasn't mentioned,
Which it wasn't.
if C1 was given to us,
Yeah.
could we also assume that the other one was C2 because it wasn't given to us and it's what's different from C1?
No.
No? Okay.
Yeah.
So we should just go with what's given to us?
Yes. And if anything's missing, then you can decide, say, I'm assuming that this is the unstated genotype. Make it up if you want. Okay, so if that genotype, if that's the full genotype, there's one individual, there's another individual. Is that a test cross? So if it's a test cross, what are the two different genotypes that are used in a test cross? One is homozygous at every locus, that's the tester. And in this case, what's the other name of the genotype that you have to cross the tester individual to? So in this case, it would be we're talking about 3 genes, so it would be a trihybrid. Meaning, in this case, heterozygous at each locus. A1 over A2, B1 over B2, C1 over C2.
But the first one isn't homozygous.
Right, so neither of these individuals, the ones circled in red, neither of those genotypes are either. Neither of those genotypes is homozygous at every locus, A, B, and C, and neither of them is heterozygous at every locus, A1, B1, or A1, A2, B1, B2, C1, C2. So that is not a test cross. Neither of the genotypes are the ones you'd expect for a test cross.
Do you need both of the characteristics of happening, or can you have like both homozygous, or both trihybrid, or one homozygous and the other one trihybrid?
Test cross is always fill in the blank hybrid, monohybrid, dihybrid, trihybrid. Has to be heterozygous at every locus you're looking at, and the other individual always has to be homozygous at each of those, for each of those same genes.
So those two steps have to be there to be a
Yep,
test case.
that's correct. Yep, both of those parts 1 and 2. One parent has to be homozygous at each locus, the other one has to be heterozygous at each locus. Good, thanks for clarifying that. Now, since we're out of time, hang on, I've got one or two brief announcements. Please feel free to work on these other questions if you want as practice for the exam. They're good ones, especially the second one. Focus on the second one first. Please do, I'm going to post one relatively brief YouTube video sometime this evening to watch for Monday. And then finally, we're going to get around to answering this question that I posted a couple of classes ago. And here comes your Socrative exit activity. Again, one question, or maybe it was 2, but they're quick. Okay, one question.
So the answer is no, it's not a test cross, because it doesn't satisfy the first one.
Correct, it's not a test cross. Okay, there you go. Thanks all for your attention. I hope you all have fantastic weekends. Enjoy the weather, and I'll see you on Monday.